Hi everyone, good afternoon. This is going to be the last set of notes for exam 2 from the professor for the applied micro exam. That's next Tuesday. So vaccines, we already talked about vaccines, and I took Gen Micro. There's a whole note slide on vaccines, but this is like a brand new set of notes for the class I'm in right now. So we're going to go over again just the slides to have that icon. Okay, so we start with antibody properties and the functions. So antibodies could neutralize toxins or viruses. This is where you're going to have the virus and the antibodies are just going to surround it like that. The virus can't bind to anything because of the antibodies. Same thing with the toxin. Opsonization, this is going to be the enhanced phagocytosis of a bacterium. So the bacteria is going to be coated in the antibodies, and then the phagocyte, it's going to have this enhanced uptake. Cross-linking, this is when you're going to make an aggregate, so you have bacteria, and the antibodies are going to make like these cross-links with the said bacteria. So something like this. Immobilization, this is going to be similar to cross-linking. However, the bacteria are going to be taken by the antibodies by their flagellum. The flagella are going to be held down by the antibodies. Complement activation, this is going to be using the classical complement pathway, where the antibodies are going to be found by the C1q protein, and then that's going to cause a cascade, and then you could have opsonization. We also have antibody-dependent cellular cytotoxicity. This is going to be using the natural killer cell. The natural killer cell is going to see the infected self cell, and the infected cell will have all the antibodies on the outside. The natural killer cell will recognize that, and it will kill that self cell. All right, let's talk about IgG, the production of antibodies. So you have 2 heavy chains and 2 light chains. Okay, so we're going to draw this right here. You have the antigen binding site at the top, and then we're going to have, of course, our heavy chain, which is this long piece. Remember, it's kind of like in a Y. The light chain will be up here in the FAB region, or the also known as the variable region. So this is going to be the light variable. And then also in the FAB region, or the variable region, you also have a heavy chain. So VH. At the ends of the variable region, you'll have the antigen binding groove area. The heavy chain will extend down into the constant region. This is FC, the constant region. So you know in Y, where you have those 2 roads and then they come together at a point and then downwards. That downwards portion is going to be the constant region. That's only going to be the heavy chain. All right, so we're going to have these disulfide linkages between the sulfurs in here. The N-terminal ends of the heavy and light peptides will form those binding sites. Again, this is the antigen. And at the end of the variable region, you're going to have the binding sites. The amino acids at these binding sites are very hypervariable. There's an antibody for every possible type of antigen. So there's a lot of variation here. That just comes from a bunch of recombination, all these junction diversity events and whatnot. So again, a lot of variation at the binding site. So you virtually have an antibody for every possible type of antigen. So let's talk about killing invading microbes next. So first you have non-specific immunity. Think of the innate immune system. The surface molecule of the pathogen is recognized. This could be a PAMP. And it's going to be non-specifically ingested by the this is wrong. It's going to be non-specifically ingested by the phagocyte. this is wrong in the slides. It says pathogen twice, but it's supposed to be ingested by the phagocyte, not the pathogen. All right, specific immunity. This is going to be like the adaptive system. The antibody will bind to the specific epitope on the pathogen surface, and this is going to allow them to then stimulate a macrophage to take care of that that pathogen, and the macrophage is going to be stimulated by the Fc region of the antibody. We also have complement. Complement, again, this is where C1q is going to be activated, and you're going to make this membrane attack complex. The pathogens can be killed by binding the antibodies through opsonization or the molecular attack complex, the MAC. Let's talk about cell-mediated immunity next. The antibodies have no effect against microbes inside the host cells, so no effect against the intracellular microbes. The T lymphocytes, therefore, are the next line of defense. These are kind of the eyes inside the cells to see what's going on, if there's any intracellular pathogen. The T lymphocytes are going to recognize the viral proteins or the antigens on the surface of cells infected by microbes. So, again, the surface of the cell will express these MHC complexes. So the CD8 T cell plus MHC1 will recognize the antigens on the surface of the cell. The antigens will be displayed on this MHC. When there's the presentation, and the CD8 will recognize the antigens on that surface of the cell with the MHC, this is going to lead to the proliferation of the antigen-specific T cells. And then, of course, you could have cytotoxic T cells, you could have T helpers. But it looks like it got cut off, so I'm going to have to move to the next portion. Okay, so here's an example of the T cell and B cell cooperation in producing an antibody. B cells make antibodies without T cells in some circumstances, but when you have that T cell also in the process of the B cell making the antibody, it kind of like just enhances that differentiation into the plasma cell and then the antibody. So the vaccine, so let's say the vaccine has a polysaccharide antigen with a carrier peptide. That carrier peptide, more on that later. The vaccine will enter the antigen processing cell, also known as a macrophage or dendritic cell. And then you're going to degrade the carrier protein into peptides that are presented on the MHC. This is going to be class 2 MHC, but it just says MHC there. The specific T helper cell, that's TH, will find the peptides of this MHC. And this is, again, the MHC is on the APC, or the antigen processing cell. The B cell requires 2 signals for activation. The first signal for the B cell will be the antigen needs to bind to the specific B cell with a receptor to the antigen. The antigen, the protein are internalized, and the peptide is displayed on the MHC. So they need to basically specifically bind to the BCR. And then you're going to internalize that peptide and then display it on MHC. B cells also display the MHC. Or number 2, this is the second signal that the B cell requires for activation. The recognition of peptide by the T helper alongside MHC. So recognition of the peptide on the MHC by the T cell and the activation of that T helper. All right, let's talk about the T helper cell dichotomy. There are 2 types. We have TH1. This is going to be secreting something called the interferon gamma. And the TH1 that secretes interferon gamma, this is going to result in local inflammation as well as the local macrophages being activated. Then you're going to also stimulate complement. You're going to be making those complement-fixing antibodies. You're going to be stimulating those. That's our next point here. Th2, this T helper 2 secretes interleukin 4. Interleukin 4 will be stimulating IgG for neutralization of toxins. And you're also going to stimulate IgE, which is important in allergy since it reacts with the mast cells. Okay, let's talk about types of vaccines. We have traditional vaccines. The first type of the traditional vaccine would be the live attenuated. This is going to be a bacterial or viral strain. It's the non-pathogenic version of it. Or you could have the killed vaccine. This is the whole cell or the inactivated toxoid. Antigens are notated as the antibody generators because if the BCR finds its specific antigen, then it becomes the plasma cell. That B cell becomes a plasma cell, and then it shoots out the antibodies. So therefore, the antigen is actually known as an antibody generator. Again, an antigen is just any molecule that elicits the adaptive response, and you're going to also have the proliferation of lymphocytes. A protective antigen. This is something new. A protective antigen protects against a later infection. Let's talk about vaccine problems. This isn't going to be tinfoil conspiracy hat nonsense, mind you. And we're going to actually just look at the types of the vaccines and then some of the history with some of their processes. Okay, so the oral Sabin vaccine, which was a live vaccine, we could have something called vaccine reversion, where the vaccine will produce one case of polio out of 520,000 people. And then the growth and tissue culture can introduce viruses from the host cell, and this could cause the tumor virus to be introduced, which you do not want. The attenuated pathogens can produce severe diseases in immunocompromised individuals. The reason why the live vaccine has this strong response, it's because it hangs around more. You know, the organism is dividing and doing that. So it's around more, so there's more of a chance of the response of the body against it. Okay, so killed vaccines. Killed vaccines could sometimes cause severe reactions. An example would be the pertussis vaccine, which is a whole killed bacteria. The gram-negative killed vaccines are going to have, of course, in the gram-negative bacteria, you have the LPS, which is an endotoxin, the lipopolysaccharide. Production of pathogens for vaccines puts the workers at risk, and the organism in a whole cell vaccine may still be alive if it's not treated correctly. Pertussis vaccine history. So before vaccination, there were 270,000 cases, 10,000 deaths from those cases, 45 million cases worldwide, 400,000 deaths. So before vaccination, a lot of cases, a lot of deaths of pertussis. The initial vaccine of pertussis would be the whole heat-killed cell vaccine of Bordetella pertussis and the inactivated culture supernatants. Those supernatants are also in there. The vaccine caused some swelling, some redness, and some fever. And therefore there's the drop of immunization in Japan and Sweden, as well as infant death and brain damage. So this technique for this vaccine at the time wasn't very refined, so therefore there are reports of the sudden infant death as well as sudden brain damage. And so Japan and Sweden stopped the usage of a whole cell vaccine. This was again the preliminary, so it wasn't like as refined, it wasn't as researched. But now the safe version of the vaccine, the acellular pertussis vax, you're going to have the contained purified pertussis toxin and the protective antigen for it, which is the same the pertussis toxin. It's going to be a protective one, so it helps you fight away a later infection. There's less fever with this one. Now we have the most revised version here in the notes, called the Chiron. Chiron now produces the recombinant pertussis toxin vaccine that has 2 specific alterations. And this inactivates the pertussis poison without affecting the protein's conformation. It's not causing a conformation change, it's just inactivating the toxin. And again, that's a recombinant pertussis vaccine. Alright, this is how they made the hepatitis B surface protein with the plasmid for that vaccine. So DNA for the surface protein, the S protein, was inserted into this YEP plasmid. So let's go and write that first. Let me find it. I don't see it in the picture though. And so anyways, it's behind the effective yeast promoter as well as before a terminator. So the S protein is behind the promoter of a yeast and before the terminator. Okay, let me try and find that on here. I still don't see it. Oh well. Okay, so surface protein SAG, which is the surface antigen, and the core antigen were also involved. They're in the plasmid. They're in a plasmid. Can't find this one either. The SAG is removed by restriction enzymes, so we're going to remove it by restriction enzymes. SAG, and then you're going to insert it into PHBSS, which is another plasmid. Let me try to find that. Okay, I found that. Alright. Okay, so afterwards you're going to remove... Oh, wait, no. You're going to then insert it into PMA56, which is another type of plasmid, behind an alcohol dehydrogenase promoter. Okay, so the plasmid also contains the 2-mu origin, so it could replicate in yeast cells. Okay, so there's a whole picture of that. I really can't explain it, but basically, you construct the plasmid by removing the SAG by restrictions, and then you insert it into PHBSS. Then you remove it and insert it into PMA56 behind the alcohol dehydrogenase promoter, and then you also add that mu origin, the 2-mu origin, so it can replicate in the yeast. There's a diagram, but yeah, let's keep going. Okay, subunit vaccines. Advantages: they're effective, they're easy to produce, they're safe, they're inexpensive. They don't have any of these components that will cause side effects. The problem would be if the current vaccine is effective, aka it doesn't have any problems, there won't be any improvements into it. But we always need to make improvements into vaccines, because they won't spend money on the testing, unfortunately. The manufacturers won't spend. Low level of expression might also be a problem. The proteins will have incorrect folding in E. coli. The growth in the cell culture lines requires tumor-inducing DNA to be removed. Again, we need to remove the tumor DNA. And these might not be as effective. Alright, let's talk about improving the subunit vaccines. So we're going to now be talking about improving them. Okay. You're going to produce a concentrated array of proteins. Methods would include to fix a large number of the antigen onto the surface of a particulate carrier. You could also use insoluble aluminum salts as adjuvants. There's also the locally high concentration of immunogen on the surface in that situation. There's also the oil-water adjuvant, and the oil-water adjuvant of squalene, Squalene, also known as pluroric acid. I'm going to highlight that. This amphipathic antigen will insert to the surface partly to create a two-dimensional array. The adjuvant, again, is just a substance that enhances the body's immune response. Let me add that aluminum salt. So there's a local high concentration of antigen or immunogen on the surface when you use the aluminum salt adjuvant. Okay, so let's talk about live attenuated vectors. There's a strong immune response by eliciting the incorporation of the subunit antigen onto the live attenuated organism or particulate, because we could also have the live attenuated virus. Okay, so you're going to be using the incorporation of the subunit antigen and the live vector. This is going to present the antigen in a natural means. You're going to then have a stronger immune response, and you could also use smaller doses. However, this could actually still revert. Remember, live vaccine could still revert, or it could act as a virulent strain in a weakened host. So again, that idea of immunocompromised people. Okay, the mRNA vaccine. Let's get to this. Okay, so the benefits of the mRNA vaccine. Since it is just the nucleic acid, safety, it doesn't integrate. That's another one thing right there. We're going to get rid of all those conspiracy theories. It's a nucleic acid vaccine. It won't integrate. It's just a string of those, the nucleotides, basically. That's what's being inserted. Okay, mRNA will be non-infectious as well. mRNA is degraded by normal cellular processes. The half-life could be regulated using modification and delivery methods. The immunogenicity of the mRNA could be down-modulated to increase safety. So you could always down-modulate the immunogenicity of mRNA to increase safety. Efficacy, the various modifications to mRNA for its stability could also make it highly translatable. You're going to have this efficiency in vivo delivery, formulating the mRNA into these nanoparticles. You're going to be using nanoparticles for rapid uptake and rapid expression in the cytoplasm. There's also the notion that mRNA is the minimal genetic vector. Therefore, the anti-vector immunity is avoided. Production, mRNA vaccines have rapid, inexpensive, and stable manufacturing. Scalable. Sorry, not stable, but scalable. Okay, so these are just the modifications. You add the cap to recruit the ribosome, the 5' UTR. You add the signal to direct the peptide to the ER. Then you have the protein, the spike protein mutant. You have 2 stops at added into there to the 2 stop codons. You have 2 prolines to modify. You need to modify the prolines for the correct configuration, and then you have the 3' UTR. That's against stability. Poly A tail, even more stability. You change the bases to G's and C's whenever there's that wobble. You remember in the amino acid code. Okay, COVID-19 vaccine production steps. So first, the DNA of the spike is engineered in the plasmid. Then E. coli will be transformed using that said plasmid. Then you grow the E. coli in the bioreactor to increase the number of plasmids. Then you isolate the plasmids, you recover E. coli, and you cut the spike protein out of the plasmid, probably by a restriction enzyme. Then the dsDNA strands are separated, and you use transcription. You're going to make mRNA. Then you move the mRNA into the nanoparticle, and you have the COVID-19 vaccine. All right, so just another bit of trivia. This isn't going to be on the test, but Pfizer one has messenger RNA. You have salts, sugar, and lipids. Moderna, you have mRNA again, stabilizers. Then you also have lipids. Johnson & Johnson, J&J, has the recombinant adenovirus in it, and this adenovirus has the instructions basically for the COVID spike protein. That's how you're going to deliver it. Viruses are very efficient in carrying, they're like little packages of genetic material, where you could change that for something useful. You could, again, put the mRNA in there if you wanted to for the spike protein. So then your body actually is the factory to stimulate the immune system, not the vaccine itself. Your body does it when it's the mRNA. Okay, you also have stabilizers, salts, and pH balancers in Johnson & Johnson. All right, that's going to be all the notes for the vaccine version of the slides. And the test is Tuesday. Please do something nice for someone, and have a good one.